To introduce Dr. Nathan Sniadecki as our microsystem speaker today. Nathan comes to us from the University of Washington, where he's an associate professor in mechanical engineering. I suppose I really should say that he rejoins us because Nate is a graduate of the Department of Mechanical Engineering here on campus. In fact, I'm very honored to say that he was a lab member of my own group. He did his dissertation work in the topic of modulating electroosmotic flow inside of thermoplastic devices. Of course, like most really good grad students, he decided to go in a completely different direction when he graduated. He joined Chris Chen's group up at Hopkins in
0304, yeah.
And was with Chris at Hopkins for about a year, maybe 9 months before he went up to where he shifted up to Pennsylvania to UPenn. So Chris Chen's group works on a number of things, but Nate's involvement was on biomechanics of cells. And he's carved out a very nice niche for himself in that topical area now at Washington. I think I'd be remiss if I didn't say a couple things about the many awards that Nate has won. I need my notes for this. So he's a recipient of the National Research Service Award during his postdoc work at Hopkins, received a Harvard Fellowship at Penn, recipient of a NSF Career Award, DARPA Young Investigator Award, Young Faculty Award, and recently was named the Albert Kobayashi Professor in Mechanical Engineering at UW. So without further ado, we are here to hear about getting a grip. And I'll leave it to you to
Thanks. No, thanks for the introduction. It's a real thing. It's a real pleasure to be back here. Actually, some of the work we're talking about today is some microfluidics as well, so I've kind of come full circle, which is nice as well. But it's been great to kind of see some familiar faces, and I can say that all my professors have not changed. You guys all look the same, so yeah. Good job to you guys. There's something in the water here. But it's been a great visit so far, and I thought I'd give you guys a talk about a biological system, really the cells of your body and how they play a role in cardiovascular disease. So this is my disclosures. So engineering itself, I mean, I just say looking at biology from an engineer's perspective goes back many years. Leonardo da Vinci looked at the blood vessels of our bodies and kind of compared them to the flow of rivers and how older rivers become tortuous. But they didn't really have the technology then to understand what made this happen, what was changing within the body that allowed for your anatomy and your physiology to change. And it was really the cells. It wasn't really until Robert Hooke saw cells for the first time with his microscope. It reminded him of the little cells that monks lived in monasteries. Suffice to say, a lot of the work that we do kind of builds off of Robert Hooke's laws of springs, which I'll share with you guys. But we look at engineering from the biology that happens during heart development, during diseases like atherosclerosis, which is clogging of our arteries, and also our ability to form clots, hemostasis, which is a really amazing phenomenon that your blood knows how to plug holes and go from being a liquid to a solid state. So for us, we're really interested in the tools that allow us to understand the mechanics of cells. And when I'm talking about mechanics, I'm talking about the forces that your cells generate as they push and pull on your tissue to form it and to maintain its function. Now for us, we're interested also in how they have a mechanically responsive behavior. So your body is undergoing shear flow, there's stretch going on, there's different stiffnesses of these materials. All of this can affect the biology. So we want to be able to control those mechanical cues to understand how the cells respond to that. And lastly, we just don't want to have this knowledge for the sake of knowledge. We're very interested in translating this from the lab into something that's going to save lives. So we've been working a lot with diagnostic devices for improving healthcare. So when I first thought about cells, I thought of them as watery blobs that just didn't do anything. But if you actually take, for example, a neutrophil here, this is a classic video from the 50s. This is a neutrophil chasing after a bacterium here. And you can notice that it's changing its shape, it's moving around, it's shoving aside these uninteresting red blood cells here. It's able to change courses, and it's doing this by the ability to generate force. So it's generating traction forces to allow it to crawl across a surface. If you're talking about platelets, they're using gripping forces that allow them to stick and hold on to a surface. Or if you've got groups of cells, they tug against each other to maintain the integrity of that tissue. And of course, your heart beats, and it's due to the contraction of the cells there. So forces are very fundamental, and there's been a lot more interest in how forces play a role in our biology. But the problem was, there wasn't really the tools out there. Now, I want to give you guys a little background. Cells here have some amazing machinery inside that allow them to generate these forces. You have actin filaments, which are dynamic polymers that will assemble and disassemble. And where they assemble, it helps push the membrane forward. And where they disassemble, things let go. There are motors inside your cells. Myosin, you may be familiar with myosin from your muscle, but every cell in your body has some form of myosin, or I should say nearly every cell. Only the interesting ones do. Red blood cells really don't. And you also have attachment points, places where the force being generated by this myosin can be transmitted outside the cell to the surrounding area. And that's done through proteins like integrins and cadherins that allow for that inside to outside connection. So from this video in the 50s, more recently we can now look inside cells. What you're seeing over here is the actin filaments that have been modified to be green. This is green fluorescent protein. So wherever you see these filaments, this is where there are actin fibers. And acting on these fibers are these myosin motors that cause these filaments to slide past each other, which creates strain inside the cell that allows them to then control their shape or generate force. So if we run this video here, this is a cancer cell migrating. And you can watch as it protrudes forward over here. These little red spots are where it's forming those adhesion points with the glass cover slip. And there's a very dynamic structure going on here as these filaments are being pulled by the myosin. You don't see the myosin here, but you're seeing the action on the actin here. And it's pulling the cell from going from the right to the left over here. So how do we measure that? Well, back in the 80s, you could put cells on a thin layer of PDMS or silicone. Literally, you would take silicone polymer, wave it in front of a hot flame, and you would get a skin layer on it. And if you put the cells on that, they would form these wrinkles. So as you watch the cells contract or relax, those wrinkles would go away. This is very nice for your ironing board as well, to get rid of the wrinkles out of your clothing. Now this is semi-quantitative. You can kind of see how much force there is by the number of wrinkles or the length of those wrinkles, but it's not very quantitative. Later on, Yuli Wang and Micah Dembo developed a spot technique where you basically have a gel, a very soft gel, with fluorescent markers in there, and as a cell crawls across that soft bed, those little beads move. And so you can calculate what the forces are based upon that elastic deformation, that strain field. People have done more work of having these markers be a regular pattern. So what you're seeing here is a little heart cell beating and deforming the dots, which are those markers. And when I left Don's lab, I had seen the work of Chris Chen, and that really captured my eyes, which was putting cells on these cantilevers. These are arrays of PDMS pillars, which you can put a cell on top and measure how much force there is by how much these pillars deflect. So this is just a simple cantilever equation based upon the height and diameter, but you have here Hooke's law. You have a force, you have the spring constant K, and you have the amount of deflection over there. So in my lab, we've been making these to be anywhere from 200 nanometers to 3 microns or 5 microns in diameter. And we have different heights from around half a micron to even 15 or larger micrometers. So this is what it looks like under SCM. And here's a cell sitting on top of these arrays of pillars. As it's migrating, it's bending these pillars. This is a very dramatic amount of deflection over here. Usually we see much smaller deflections, but this is a very dramatic video which helps you get the impression about how much force these cells generate. So with this, we have a real direct calculation behind how much force is being transmitted at each of a cell's adhesion points. So really we have a decoupling, because with the other techniques you either didn't have the ability to quantify very well, or you were looking at that strain field of those beads moving and then trying to do the inverse calculation to figure out what was the force field that was generating that. But here we truly know how much force is being applied at each of these adhesions because each sensor is independent. So this is a really simple process here. It's just SU8. You do exposure to make a master structure out of SU8, and then you do make a mold, which then you cast to create a three-dimensional photocopy of your original structure. These are very simple to make. We make dozens to 100 a week in my lab as well, because each one's a one-shot deal. We don't want to use this over again because of the amount of proteins that you put on there. It's not very successful to reuse. So let's focus in on homeostasis. So this is a clot here, and these are platelets. These are perhaps the smallest cells in your body, but they play a huge role in your ability to form clots. So what you're seeing over here are red blood cells trapped within a mesh of fibers. These fibers is known as fibrin, and they are created by the reaction of these platelets. You have clotting factors that allow for thrombin to be activated, which needs that platelet surface. But what's rather interesting is these platelets are also playing a mechanical role. They will contract and squeeze the clot tighter together to prevent it from being dissolved away by other enzymes. So really, platelets are the most important part in stopping your bleeding because they're the first ones to recognize a wound site. So if you've got an injured vessel, your platelets will stick to the tissue underneath that blood vessel. So that's that first response to injury. They'll then aggregate together to form a very fragile plug that stops that initial bleeding. It's then this process of secondary hemostasis where you start to create that fibrin meshwork that helps to reinforce the clot and really provide it the strength to not break off due to the pressure and flow in that artery. So without these platelets first sticking to that site and also pulling and packing together tightly, you're not going to have the ability to clot, and that can be a very dangerous situation in trauma. So really, platelets are these kinds of grenades, or if you master transformers. They're initially these very small lentil-shaped cells. They're around 2 to 4 micrometers in diameter. And as soon as they encounter a wound site, they undergo a massive change in their structure internally. Normally, they have this pool of polymer pieces. These are the monomers of G-actin. And as soon as they get activated, that G-actin, sorry, it looks like I've killed this thing. I'll go over here. As soon as they get activated, those polymers start to come together, forming these filaments that can push the cell's membrane forward as they reach out to grab hold of each other and to form this clump of platelets. They then start to pull in tight with each other that allows them to now form this really tight, strong mass. So for us, we want to measure these forces that happens as these platelets come together. Because if they can't hold and grip onto each other, they're going to break apart and they're going to be more porous to allow enzymes to come in and break up the clot. So we take our microposts and we just stamp the tips with proteins. We usually can use anything like fibrinogen or fibronectin or proteins like von Willebrand factor that will absorb onto a PDMS surface. We then change that surface to become more hydrophilic with some ozone, and now we use a process called microcontact printing, where you can now bring that stamp into contact with the tips of the pillars, and you get protein to transfer across. So now the tips are sticky, and we want to prevent the platelets from binding anywhere else. So we use a material that's a PEG-like polymer called pluronic, which absorbs to the walls and to the base that will prevent any additional protein from sticking. And at that point, we can put our platelets down and see how much force is being generated. So some work we did about 4 years ago is we were just looking at the forces that your platelets generate. And so what you're seeing in this SCM image is a clump of platelets that are stuck to the tips of these pillars that are contracting inward. And we can measure how much force there is by quantifying how much those tips deflect from their neutral position. So in some cases, we might just have one platelet on the tip of each post, or if we're lucky, we get a clump of them to form together. And so you can see in that right image over there how much force is being generated by analyzing how far these tips have moved from their neutral position. And so we can use this as a way of screening different compounds that we know affects clotting. So for example, thrombin is a very potent activator of your clotting ability. And so as you add thrombin to these platelets, we're seeing that their forces increase. So that thrombin is helping to hold tight, to help these platelets hold tight to their surface and stabilize that very early clot that's forming. We can then look at some different drugs that we know affect the ability for cells to generate force. What you're seeing here are 3 different compounds that affect the ability for myosin to generate force. You have ML7 on the left, which inhibits a protein called myosin light chain kinase. You have another one in the middle called Y27, which also affects myosin, but it does so by affecting a protein called Rho kinase. And on the right over here, you're seeing results for blebistatin, which directly affects myosin's ability to act like a motor. It affects its ATPase activity. And what you're seeing in these graphs over here is as we try different concentrations of drugs, we're seeing different relaxations in these clots. So this kind of demonstrates the ability to screen new compounds if you're looking at clot-busting drugs. A lot of cardiovascular disease is due to clots happening in older people where you don't want them. So you might go on warfarin or you might go on baby aspirin. And the idea there is looking for the right kind of drugs that are going to prevent clotting when you want it, when it becomes too much, but not to tip the scale of balance too far where you completely stop the ability to clot. Oh, thanks. Is it going to work with each other? Okay. Oh, maybe I should go like this. Yeah. Yeah. On the side here. It's like green looks on. Okay. Oh, good. Okay. So as I was saying, this is a way of screening different potential antithrombotic drugs. Now what's interesting with platelets is when they attach to that wound surface of one of your arteries, the first thing they bind to is a protein called von Willebrand factor here. This is in our blood. If you don't produce this protein, you could also have a hemophiliac-like syndrome. So platelets use this protein to first bind to a wound surface. They then activate and bind to other platelets through von Willebrand factor or proteins like fibrinogen to form this connection of 2 or more platelets together that eventually leads to this aggregated structure here. They activate each other by releasing, they activate thrombin, or they release things like ADP from within their little compartments that help to signal additional platelets to stick onto that mass and to form that plug. And what's rather interesting is that this whole process of clotting starts with this first step here of a platelet sticking to a surface. And so we were interested in what goes on here. How is a platelet gripping hold of this surface here? It's not using the receptors that we normally think about when cells grip onto something. Normally cells will grip onto a surface with integrins. These are proteins that stick halfway inside and halfway outside of a cell. They hang out in the cell's membrane. And these integrins are what bind to von Willebrand factor, or they bind to fibrinogen. But this protein over here, glycoprotein 1B95, doesn't seem to have a role, or does it? So I've seen these images before where this is that protein. You've got 4 subunits. You've got glycoprotein 5, glycoprotein 1b alpha here. You've got 1b beta and 9. And together they form that complex. And this is the outside part. So it's really this glycoprotein 1b, this tall stalk protein, that binds to the von Willebrand factor and allows a platelet to stick under flow. inside the cell, the same receptor, the same protein, can bind to actin. It does through a protein called filament A, which acts as this intermediate between actin and this receptor over here. What's interesting is we know that this receptor acts like a catch bond, which means that under tension, its bond lifetime increases. So that makes sense for flow, because as a platelet's rolling along, once it gets caught, once the fish hook gets caught, it now stays in place a lot longer, because this receptor over here changes shape and becomes increases the bond lifetime by tension acting on it. What if tension could come from the inside? What if the cell's ability to generate force through actin and myosin could be transmitted from the inside to help a cell adhere to a surface? So we sought out to understand that. And really, we looked at integrins, which do the same thing. Integrins are a similar kind of transmembrane protein that can connect to actin, and it has a host of proteins that help in bridging the force generation apparatus to the outside world. Cells also have it between cells. You have cadherins, which form these homodimeric complexes with another cell that also connect to actin, and you can transmit force that way. So if these two guys can do it, why not GP1B? So people have been looking at platelet forces just a little bit. We did a paper back in 2010. Since then, they work with AFMs on single platelets, or they would put a single platelet on that polyacrylamide gel that has those little fluorescent beads in it. And so from that strain map, you could calculate that force map produced by these cells. But with all of these, you're either looking at a clump of platelets or individual platelets, but it's very hard to look at the individual adhesions or control how a cell sticks. So we couldn't use our microposts, so we had to go nano. So he's worked with a very nice e-beam lithography system on campus at UW. We were able to make these very small silicon pillars here. And from that, we did our double casting process to make these PDMS nanoposts. We actually made them a lot smaller than this, but we found that if you go too small, PDMS starts to fail in terms of the fabrication and getting the kind of aspect ratios that we want here. So this is a platelet on these arrays of nanoposts. What we first do is we coat it with von Willebrand factor on the tips, and now we stick platelets on. They will spread out and generate force. You're seeing a lot more dramatic force here because this is an image done under scanning electron microscopy. So whenever you take a biological sample, you have to dry it. And so that drying process oftentimes shrinks the biological sample. But you can see under these confocal microscopes, we do see deflection of these pillars, and we're able to measure the force there. So we did a lot of biology. This is actually a collaboration with Jose Lopez, who's at the Puget Sound Blood Center. He's got a wealth of different reagents to work with for platelets. And so one of the first things we did is we used antibodies that can block the specific receptors of a platelet. So one antibody is AK2, which blocks that target of interest for us, the glycoprotein 1B. We can also use this antibody called 7E3, which blocks the integrin in platelets. Because both of these, both the integrins and this protein GP1B, can bind to von Willebrand factor. So we needed to decouple those two responses. So what we found here was when we added AK2, the cells were not as spread, and they actually produced less force. We're showing everything here in percent to control because each person's platelets generates a different amount of force. And so what we've done here is normalize this study by donor-to-donor variability. But a little bit of this antibody here can reduce the amount of force being generated. It's not as much as if you inhibit the integrins, but this is definitely showing us that there's a significant loss in force by blocking that receptor. So we need to now confirm it with another way. This time, we just isolated the protein, the domain of residues within the von Willebrand factor that allows the GP1B receptor to bind to it. This is the domain A1. So this is just a small domain here. We use biotin and streptavidin to put this on our posts. And so now the platelets that are on, sorry, I think I've burned the second one out. I think my thumb is too heavy. So over here, what you're seeing is a platelet that's able to generate force. It's still a little small, but it's showing that they're able to transmit forces through this receptor. We also did the slightly opposite effect. We took the extracellular domain of that receptor. So we took GP1B and we only used the 300 amino acids, and we sprinkled that on top of the post to block the sites where the platelet could bind to. So in a sense, we're creating a traffic jam. So now the platelet can't bind to the same sites because you have this peptide out there blocking those sites. So when we do that, we use this GP1B alpha 300. And with that, we also see a decrease in force. Going one step further, platelets are great. There's donor-to-donor variability. But oftentimes, you need to demonstrate biology in a different context. So we now use a more reductionist approach. We started with these Chinese hamster ovarian cells where they don't express any receptors at all. They're basically these empty cells on their surface. And we program those cells to now start producing that same receptor of interest, like glycoprotein 1B. So now what we have over here is all you need is glycoprotein 1B alpha, beta, and 9. You don't need to have glycoprotein 5. So this cell is acting just like a platelet in terms of its adhesion. So when we put these cells on, oh, one more thing too, we were able to also manipulate the size of that protein. We could chop off the cytoplasmic side, so we could truncate this protein at one of its residues, at its 534 residue, which is where filament binds. So we can chop off the part that can connect to the actin cytoskeleton. So with these CHO cells, we first put them on von Willebrand factor, and we found that they were able to spread out and generate some significant forces over here. And then if we used this CHO beta 9, this is now missing one of those 3 essential protein subunits. It's missing the alpha subunit. So without that, it can't bind. And so we don't see any adhesion and we don't see any force. So this confirms to us that these platelets are not binding, these CHO cells are not binding through any other mechanism. They're only binding through this receptor that we're putting back into that cell. We also show that these forces are dependent upon myosin. We added blebbistatin, which I showed you guys as one of those 3 drugs that we had looked at earlier for platelets. When you add blebbistatin, you see a reduction in force as well. And then we also use this truncated part where it no longer has the full length of its cytoplasmic side, this delta 534 version. And here we see a reduction in force as well. So what we're seeing over here is that this receptor is able to allow platelets to grip onto a surface. So this opens up a new area for drug research. A lot of research has been done on trying to block the integrins of platelets to prevent a thrombus. Now, perhaps, they can start using glycoprotein 1B as a target for new drugs that can prevent a clot from happening where you don't want it. So this is what clot looks like in a bloodstream. So this is in a mouse model. This is not from my lab, but this is a process known as intravital microscopy. So blood is flowing down this channel over here, and you're getting a clot to form here. Red are your platelets, and you're watching as this thing grows and mushrooms into the flow stream. And what's happening is you have a shell of platelets on the outside. In the middle over here, you have a core of platelets plus fibrin that forms. That's that meshwork that holds it in. So the outside is very loose. And then the wound over here is where you have tissue factor that's embedded in. That's also a very strong activator of thrombin and clotting. But what you're watching here is there's a change in the shape of this clot. It starts off very early where you have just a bunch of platelets sticking. It grows very quickly, and then it starts to condense. And it's not just the shell. You're not losing platelets off the top. You can actually see in this purple zone over here, it's rather large and then becomes much smaller. These platelets are compacting and condensing that clot to make it rock solid. So that's an important process. You need these platelets to generate force, to grip onto a surface, and to consolidate. And so this is very important in the case of trauma. So about 5 million people die worldwide. So really, thrombosis or thrombotic diseases happen in the older generation. Hemostasis is very important for the younger generation because oftentimes you do it to yourself through accidents or other dumb behavior. And it's a big burden on the healthcare industry. It's about $135 billion in trauma-related treatments. So oftentimes, there's an accident, they have to get to the major trauma center, and a physician has to figure out what's wrong with their bleeding. Because in fact, 25% of major trauma patients can't clot. They're at risk of internal bleeding because the injury could have caused anticoagulants to be activated in their blood. They may be on aspirin and are unconscious and can't tell you that. Or there's a lot of other reasons, like hypothermia can induce this problem. So a physician that's receiving a patient at the emergency room has to figure out what's wrong with their bleeding, because they could compress parts of the body that are bleeding that they can visibly see. But if you have internal bleeding, you can't put a tourniquet on it. You can't compress it to stop the bleeding. So the best remedy there is a transfusion. But that's like an elective organ transplant. You know, your blood is one of your organs, and so you don't just elect to have a heart transplant if you need it, or if you don't need it. You know, so the thing is you need to understand, does that person need a transfusion or not? And what kind of transfusion? Should they be getting the plasma? Do they need more platelets? Or do they need something that will cool off the enzymes that are chewing up these clots? So you really need a very rapid way to detect this problem, because in trauma medicine, you have about a golden hour, they say, to identify what's wrong and then quickly give them a treatment. But the problem is, by the time you get pulled out of the vehicle, put in a rig, taken to the hospital, half the time is gone. So there are tests out there that work, but they take about 30 minutes to run that test. So already your golden hour is used up. You need a much more rapid way of doing this. So this started as a DARPA project because this same kind of issue relates to the warfighters. Roadside bombs, IEDs, created massive amounts of internal damage in our veterans. So they needed a very fast way of detecting these problems. So we thought about using our microposts. And in this case, we're using a block and post. So as whole blood flows through our microchannels, red blood cells pass on by, white blood cells pass on by. But what you're seeing over here are platelets forming a clot down on the downwind side of that block. And as they form that group of platelets, they start to contract. And we can see how much force there is by how much this pillar deflects. So in these microchannels, we have hundreds of these little sensors, and we can form hundreds of little microclots. And they form within seconds. And by looking at the force, we can detect what's wrong with the blood. If the plasma is bad, you won't see a clot forming. If the platelets are bad, they don't produce much force. Or if the enzymes are too hot, you will see that clot dissolve away very quickly. So again, this is a very simple technique with PDMS, where we now make a microfluidic device that has these block and posts in the bottom. And so there's a little video that my PhD student made that I'll show you guys. But these are very small structures. We have an array of those in the microchannel. We really do work this fast in the lab. It's amazing. You go into the clean room and out it comes back, all done. All that work is done. It's fantastic. We use buckets of PDMS here. Pour it on, get the bubbles out, cook it up, and you got your device ready to go. We then assemble that device, put the proteins on. So first we do some plasma treatment with it. This will allow us to bind the proteins that we want, like collagen. We treat it, and now we can work with blood. So we get a lot of blood. Everybody wants to give blood. It's a great thing for undergraduates. They make $15, so we have no short supply except when school's not in session. So we can take blood from a donor, and we can mix it with some different activators if we want to. We've actually done it. We've done a direct draw, where the blood is drawn and immediately tested in our device as well. But if we want to test different compounds or different treatments, we can go ahead and now inject that blood into our microfluidic device over here. We keep it at body temperature because clotting can be affected by the temperature of the body. And so it's just simply looking to see what happens in this device. And this is done on a very large microscope. We've actually now made a prototype that's about 8 inches by 8 inches. It's a miniaturized microscope that allows us to see the same effect that we see in our larger microscope. So we do this optically. We can see how far the micropillars have bent from their neutral position. So you see that on the left. And so we can look at these curves. Within 3 minutes, we can see a normal clotting response where we see the force starts after 30 seconds, rises up, and reaches a preset level. But if the blood is impaired, if something's wrong, we see a reduced amount of force. So this reading of force is sensitive to a lot of the things that go wrong in coagulation problems. So if you have impaired coagulation, maybe your plasma is not producing thrombin. So here, on the left, we used heparin to inhibit thrombin. And when you inhibit thrombin at different concentrations, going from black, red, to blue, we see a reduction in force. Or, in the middle over here, this is aspirin, acetyl salicylic acid. You can take that, you can mix it with the blood, and at different concentrations. So at the red curve, that's about taking a baby aspirin, about 80 milligrams. 0.5 is like taking 2 aspirins. And what happens is you see a reduction in the force that these platelets can generate. And on the right over here, this is fibrinolysis. These are the enzymes that can chew up a clot. So a compound known as tissue plasminogen activator, or TPA, that activates the enzyme that eats up the fibrin. So when you add that to the system, you see a reduction in force as well, because those platelets need to have the fibrin in between to connect with each other. So if you chew that up, things are going to fall apart and you're going to get less platelets stuck at that device. So we've actually done a spin out on this. This is Stasis. I'm one of the founders. Nathan White, who's an emergency medicine physician, is another founder. And then through the university, we met Bob Berry, who was an entrepreneur in residence, that the three of us had launched the company. What's nice about the University of Washington is we had a lot of commercialization support. So through the Coulter Foundation or the Life Science Discovery Fund or even the National Science Foundation, it's been a really great environment to be able to develop a technology and kind of push it or kick it out the door, but be able to have it land somewhere and really get some ground, some stable ground for it to grow. All right, let's move on to your blood vessels. So inside of your arteries, you have an inner lining, which is your endothelial cells. And these cells see flow. It's actually kind of amazing. If you were to take all your blood vessels and open them up, that inner surface area is as big as 1 acre. That's a lot of cells. And the thing is wherever there's a defect in that inner layer, something can leak through. So if there's a cut somewhere in that giant 1 acre, a platelet can stick there. Or if you've got cholesterol, LDL can leak through into your arteries and now form a plaque. So you don't want there to be any gaps within this group of cells. You want them all to hold tight and form the best patchwork quilt that you can imagine. So we were interested in the ability that these cells have to pull against each other. So this is a study that I had done when I was a postdoc, where we put cells as two, a pair. and we measured the tugging force between them. What we found was that these guys are able to generate a tugging force, and the amount of force that they generate is proportional to the amount of glue in between the cells. In this case, you see a stain for beta-catenin, which is a protein that helps form the adherence junctions between cells. So the bottom line of this story is that the more the cells pull against each other, the more glue or stitching between the cells. So in the case of endothelial cells, you want these cells to pull tight against each other because that's going to help form a very good barrier because you're going to have a lot of beta-catenin and other proteins that stitch everything together. So in my lab, I was interested in how does this phenomenon change under flow. So what's interesting, if you look at arterial flow, you have regions where you have laminar flow, where the blood flow is steady, but you have also regions where you have disturbed flow. It's not really turbulent, it's oscillating, it's unsteady. And you have different behavior in the cells in those two regions. So on the right over here, you're seeing a small capillary, and those endothelial cells are forming that barrier. But you don't want leakage. So if you have any gaps between those cells, things can leak out. What's rather interesting is that in the regions of laminar flow, you have very little plaque formation. So there's very little leakage from those regions. So there's very small gaps between the endothelial cells. Could it be because there is higher tugging forces between these cells that are helping to glue the gaps together? Under disturbed flow, it's a lot more leaky, there's a lot larger gaps, and the hypothesis is could the tugging forces be weaker there? So we built a bioreactor. We grew these monolayers of cells on our pillars, so that way now we can measure the tugging forces between these cells. And we gave them disturbed flow by using this backward-facing step, and we used laminar flow downstream. So what we found was, under laminar flow, what you see across the top row here are a bunch of endothelial cells, and across the top, they're a lot more contractile. They have more filaments, and on that bottom row you can actually measure you can see the forces that they generate. So in that middle group right there you see a lot more vectors, which means that they're producing a lot more force. Under disturbed flow on the right, they're more relaxed. A lot less force is being generated, and there's a lot more gaps between the cells. So we wanted to quantify that. So just using some calculations, we could figure out the intercellular force of one cell being pulled on by its neighboring cells. It's really due to an imbalance of that force. And we subjected these cells to either laminar or disturbed flow, and we saw that laminar flow increased the tugging force between these cells. And also, in the second column, we're measuring how much of that beta-catenin is in between the junctions between the cells. And what we found is by laminar flow, you see an increase in that adhesion proteins between the cells. But you don't see that with disturbed flow, or those red graphs. You see a lot less tugging force and a lot less beta-catenin, or another protein called ZO1, which also regulates the junctions between the endothelial cells. So what it looks like is if the flow has an effect on the ability of these cells to contract. And by being more contractile, they're able to pull against their junctions and undergo a mechanotransduction response where they help reinforce that site with additional proteins. And that may be one of the reasons that helps to maintain the junction or the barrier function of your endothelial cells. All right, one more focus point is the heart, so cardiomyocytes. So these are the cells that are in your heart that beat every second, 24 hours a day, 72 years, hopefully, or longer, and they don't take a break. So what the structure here of a cardiomyocyte is a lot of actin and a lot of myosin. You can see it over here in these electron microscopy images. This dark band from here to here is made up of myosin filaments, shown over here in these red filaments. And over here in this lighter region, from this Z line up to about the M line over here, that's made up of actin filaments. And so by myosin walking along actin, you're getting a shortening effect. And this is what's happening in every muscle, whether it be cardiac cells or skeletal muscle. And in non-muscle cells like platelets or endothelial cells, you don't have as much organization to that structure. So these are heart cells from a neonatal rat that are beating on our microposts. We're able to see them contract. We're not giving them any electrical stimulation. They're undergoing spontaneous beating. So we can quantify each twitch that goes on in this process here on a per cell basis. So one of the first questions we asked was, what's the roles of mechanics here? We know that heart cells see a change in their environment. The environment becomes stiffer. So from an unborn baby, a prenatal, the tissue of the heart is around 12 kilopascals, which has a shear modulus of around 4 kilopascals. And within 3 days after birth, as a neonate, it goes up 3 times. So there's a stiffening effect that coincides with the increased need to be a stronger heart. So in the world of tissue engineering, if we want to make better heart cells that can be used for therapeutics, we need to figure out ways to make these heart cells stronger. So could we use these mechanical cues as one method? So we did a work where we put those cardiomyocytes on different stiffness micropillars. By changing the height of the pillar, you can change its stiffness. So a tall post is soft, a short post is stiff. And so we can control that modulus of the environment by changing the height of our pillars. And what we found was these cells adapted to their environment. From the same batch, we seeded these cells on different stiffness environments. And they were able to generate more force as they were on a stiffer environment, almost, is that 3, 4, 5-fold difference in force by changing the stiffness. And they were able to generate more work and more power. So this is a very nice bit of data here, but what we're working on right now is looking at this for stem cells. So we have a collaboration with Chuck Murray, who's at UW, who works on taking adult cells. You can then reprogram them to be induced pluripotent stem cells. So you can get a very ethical source of stem cells from somebody. And then giving these guys the right cocktail of food, you can teach them or encourage them to become heart cells. And now you have a very renewable source of replacement cells that can go into a dead region, because the heart works nonstop and doesn't have time to regenerate itself. So you need to have a new source of cells to go back in there to recover the dead zones, because those dead zones are not contracting. And so the heart has to work harder in its other regions to compensate for the guys who aren't doing anything. So with Chuck Murray, we're using our microposts as a test bed. As different techniques are being developed, we can then measure the power of these cells, the force, the velocity, the power, under different conditions. So this has been a real game changer for Chuck's lab, because now they have a direct readout on a per cell basis whether something works or not. Because these are they're kind of rare cells. They're very weak, and we need to get them much stronger. So what you're going to see over here is a single stem cell that's been transformed into a cardiomyocyte beating on the microposts. And some of the studies that we've done is we've tried different matrix conditions, different proteins for these cells to adhere to see which ones help to promote stronger cells. On the bottom down here is an example of one of our studies where Chuck's lab was interested in a thyroid drug. Thyroids oftentimes lead to enlarged hearts. And it's also very important in developmental biology. You have thyroid hormones that are released to help promote the strengthening of a heart. Can we use that same hormone with these stem cells? What you're seeing here in red is a cell that was treated with a thyroid hormone, versus in black, it's just an untreated cell. And we were able to actually almost double the force by giving them this thyroid treatment. We're doing some additional studies with some siRNA or different fatty acids, but this platform right here has been a real help in looking at cells and how they respond to different cues that help to promote their maturation. So the last thing I want to talk about is an undergrad, Thomas Larson, who was in my lab. We had an issue. We had everything that we were doing on this giant microscope, and if we ever wanted to commercialize something and move it out of the lab, we needed to have a different way of looking at how these posts deflect. We tried magnetics. It was a big issue. We're still working on it, but was there an easier approach? And I asked Thomas, can you just make me a microscope out of PDMS? And it was a very simple idea. Can you just make a hemisphere? So a simple little hemisphere on PDMS that you can stick onto a cell phone can allow you to see red blood cells or really mini cells of any type. And this was a great project for an undergraduate. So great, in fact, that he said, I don't want to go work at Boeing or other places where mechanical engineers go. And I said, well, what do you want to do? He's like, well, I think I want to start a company. I said, let's go for it. So he was able to spin out microphone lens from my lab and did a Kickstarter. And within his first year, raised enough for a good salary for himself. That's just a 15x lens, and you can buy this on Amazon. I don't get anything, don't worry. But he then launched a second Kickstarter where he now has 150x, and now he's just after some delays, he's now actually sending this out to people who have backed him. And he and I are actually going to be putting in for a Bill and Melinda Gates Foundation award to try to use this in underdeveloped nations where they can use it for remote microscopy, looking at different infections, maybe in urine samples. But basically having telemedicine and having the kind of equipment that you would need to go in the field.
You can buy them on Amazon?
You can buy the 15x on Amazon. Yeah, or directly from his website. Yeah. So this is a nice, a little cute thing that can happen with PDMS. I probably shouldn't say it was PDMS. It's probably. So anyway, I just want to kind of close up over here so we have some time for questions. But I showed you some work with micro and nanoposts that we're using to understand platelets and how that plays a role in bleeding. We have this block and post device that can rapidly look at coagulation and look at that for trauma patients. And then I talked about some of the biomechanics of your blood vessels and how we're using these mechanical engineering approaches and system approaches to look at how heart cells develop. So really, I think the impact here is by having these new technologies, you're able to now understand biology from a different perspective. We have new receptors that we're understanding. We have new, faster results for medicine. And we're also looking at how mechanics plays a role in disease progression or how we can use that for actual therapeutics with heart cells. So really the whole kind of motto here is that with these new tools that can come from BioMEMS, you have these new strategies that they can be pursued for diseases and for medicine. So I'd like to just acknowledge my lab as well as our funding agencies, and I'll take any questions.
Any questions for me?
Thank you for the excellent talk. The coagulation sensor, did you observe any issues with measurements with different, basically different sources of people, and did it affect the evaluation?
No, actually, so it's not. It's interesting. It's mostly the people are the wild card. We find more of donor to donor. We've done side-by-side tests. There's I guess if you have a bad batch of one protein, but we don't really find that at all. It's usually the donors. Each person has a different nature to their blood and how strong their platelets are or what kind of proteins are in their blood as well. So unfortunately, we would love to have the same donor come back again and again, but that we can't draw from the same well too many times.
Questions. Thank you for the good talk. It was very interesting. When you're measuring the force, the deflection, is it just optically? Have you done other things?
Yeah, it's mostly optical. We've tried exploring some other ways, but anytime you're in a wet, conductive environment, you lose out on a lot of different physics. You can't really do too much electrical in there. It's too conductive. So there's been some other stuff. We've done a lot with magnetics as well, but there's some noise issues going down there. But optics, at first I was very reluctant to think about technology that would leave the lab. I didn't think optics would work, but as a PI, you're always wrong. So your students can prove that the ideas were off, and they showed that they could take a simple web camera, flip the lens around, and now they got a microscope.
And when you're putting a cell on your posts and then the are treated so that the cell wants to adhere to them, right? When you see deflections, how do you know that is the cell actively gripping and pulling the post rather than it passively just being stuck to the post, which might be mechanical elasticity or modulus or something?
Yeah. So the cell, let's see here. If you were to squeeze a cell down, it would push the post away. If you don't put down any of the proteins on the surface, the cells will not attach. They don't stick, okay? There is some very interesting questions that you kind of touched on a little bit about what receptors are involved. For us, if we've been trying to understand some of the different proteins that connect the inside of the cell to the outside. You saw a little bit about that with the work on platelets. But I think it's not all just actin and myosin that's generating force. The membrane of a cell has membrane tension, and that could be playing a role. But regardless of where that tension is coming from, it has to go through that adhesion point, because if you don't have the stickiness, you don't see force. Yeah. Yeah. But there's many other mechanisms in cells that can enable force or strain or changes in shape, and I think those are areas that we're going to continue to look into and understand. Look, another question right back there.
I think you're touching on it, but is there a concern that you might be overestimating the force that cells apply because if what they're gripping is deforming more than it would in a normal endothelial environment, that you're missing some sort of canopy that would normally stop the cell earlier than you see it?
Yeah, so I guess your question is what's the difference between in vivo versus in vitro studies, And that's, you can't kill that problem, right? In vitro is a reductionist approach. In vivo is too complex. I can't control every variable. So what we see in vitro is definitive for that system, right? And we can interpret those results to the real life, or the wet life, if you will, the biology life. There's always issues between what is an artifact environment versus what's a real environment, but I think these are the same, it's the same mechanisms, the same machinery of the cells. It may be sensing it a little bit differently, but the inner workings, these platelets are forming clots that are similar to the clots that you saw in that blood vessel under intravital microscopy. It balloons out and you have all these platelets. So I'll leave it like that.
Yeah, we have time for a couple more questions.
When you measure or quantify the forces that is reflect from the deflection of the posts, I see some of the posts are deflecting more, some of them deflect less. So what was the mechanism that you quantified?
Yeah, so the force is being measured just through MATLAB. You can look at images and quantify the position of the tips. The reason why they're deflecting less is really due to the forces the cells are generating. So you might have seen it in that one of those early videos I showed of the green fluorescent protein actin. You have different connections at different adhesion points. So the cell has a skeleton on the inside where you have different connections to different adhesion sites. So the reason why you see more or less force is because the force that myosin is generating, or many myosins are generating, gets transmitted differently through the structure. It's a more of like a web of interactions. If you have a spider sitting in a web and it starts to pull on the web, you're going to see different strands pull differently because it's a network of connections. But myosin is not just one spot, it's everywhere throughout the cell, and you have different domains of where is it more activated than other spots.
It's just somewhat related to those questions. in your micro and nano array post for the sensor, how did you determine the spacing between each post? Did you find like optimized?
Yeah, so there's always the issues. You don't want the cells to fall straight through, right? So you want to make sure that the spacing's, it's really a lot of trial and error. Oops, we made it too far, now the cells can't spread. Because of the process of spreading is they have to polymerize their actin and push that membrane out until they reach the next spot. And if you ever watch them under a microscope, they send out more of a filopodia, like a very thin fiber, and they kind of wave it till they find something that sticks, and then they reinforce that site and spread out. So a lot of trial and error on that. Too close together, and you have fabrication issues because PDMS is very soft, and as soon as you peel it, they snap together, and once it's stuck together, you really have a hard time taking it apart.
Any other questions? And I'd like to ask one
All right.
question, if I may, since I think we saw a few minutes. I'm curious, actually, that you didn't talk at all about the role of the bilayer membrane of the cell.
Oh, yeah.
As part of the chain of force transducing components of the system.
Yeah.
I guess I'm particularly, so I'm curious about that in general, about what the role of the membrane is. But in particular, so you talked about the tight junction proteins
Yep.
and the expression levels and how they change in the presence of the unsteady flows. And so you mentioned, some like the cellular occlusion.
Yep. Yeah.
The expression went way down for that protein.
For disturbed flow, yeah. Yeah.
Flow. So a lot of these proteins will organize on the membrane on the basis of the structure of the membrane itself.
Yeah.
And the structure of the membrane is also impacted by the flow conditions. So I'm curious if that question has ever been asked.
Yeah, usually people talk about flow, they talk about how is the cell sensing that flow, right? So there's this whole, how is mechanotransduction going on? So in the case of like the tight junctions, we're not focusing on how the cell is detecting that different flow. We're focusing on what the response is. But the membrane does have very dynamic properties. You've got assembly of rafts that form to kind of bring these different proteins together that are in the membrane. So I don't know, in the membrane itself, there's a mechanism called these stretch ion channels. So if there is shear flow acting across the membrane, oftentimes you can get some increase in tension that could open up pores, and that could lead to a signal event inside the cell. But you're right, the membrane is a beautiful structure with lots of interesting assembly components to it that I think in my world it's understudied. So I think there's a lot more that can be done in understanding how the membrane plays a role in cell mechanics. It's just very fragile as well. Yep.
Okay, let's thank Dr. C. One more time.